seconds.
Hello, good evening, everyone. Welcome to the last session of Epigenetics presented by Genesis, the Life Sciences Society of Shivnadi University, and KaryoUS. I hope you are all enjoying the session so far and learned a lot because we did from you all. And so, just wanted to start with the session. I think Sandhya and Shani may continue. And in case you have any doubt, you can just unmute yourself and or put it in the chat box. We will be answering it. Thank you.
Right, so let's begin the session by discussing the assignment that was given to you in last class. So, in the question, it says that there is a hypermethylation in the P53 gene in chronic smokers, and you were asked to answer what this means. So, like we have learned in previous classes, methylation causes the silencing of a gene, which means it represses the activity. So in this case, the P53 gene is a tumor suppressing gene and it suppresses cancerous tumors. So if there is hypermethylation in the P53 gene, it would mean that it cannot express itself and suppress the tumors. So in chronic smokers, since there is no suppression of these tumors, we see cases of cancer more prevalent in smokers as compared to non-smokers because there is no gene that can stop their activity. All right, so those who answered it correctly have gotten it right, and those who couldn't, I hope you have learned the answer now. So let's move on to a new topic in our discussion, which is
Shani,
epigenetic. Yeah.
sorry to disturb. Just present it the PPT. All right, thank you.
All right. Okay. So before I begin with epigenetic therapy, let's revisit the basics a little bit about epigenetic regulation of gene expression. Now, I want to ask you what you guys remember about epigenetic regulation. Do you guys remember the mechanisms of epigenetic regulation which were taught in the previous classes? Like, how does epigenetics control the regulation of genes? You can unmute or answer in the chat box. Anyone? All right, I'll continue. So, there are essentially three mechanisms of epigenetic regulation which is seen in our bodies. The first one is DNA methylation, which I just talked about in the assignment. Then there is histone modifications, and the last is gene silencing via microRNAs. Now, this machinery, these mechanisms are controlled by certain regulators. So, the regulation is controlled by regulators or enzymes in this case. So, we have learnt about enzymes in the previous classes like histone acetyltransferases, DNA methyltransferases, and histone deacetylases. So, these enzymes, these molecules, are known as epigenetic regulators. Now, this is a very consistent machinery, and as we know, if there is an alteration, if there is a sudden aberration or change in a consistent process or machinery, there are always repercussions or changes that we see. So, even in this case, alterations in this epigenetic machinery causes problems which we know as diseases, and the most notable of them being cancer. Now, when these regulators, if these molecules are altered in some way, this disrupts the epigenome, and this phenomenon is called epigenetic dysregulation. And this is what plays a causative role in disease pathogenesis. Now, how is this knowledge of epigenetics utilized in therapy? How do you think that epigenetics can be used to make drugs which can possibly be treatments for diseases? So, as we have learned that epigenetics is essentially the addition or removal of certain molecules to and from the DNA. So, in a sense, it becomes a reversible phenomenon. right? So the plasticity and this reversible nature of epigenetic modifications is the basis for developing um, anti-cancer drugs or epigenetic drugs. So this principle has been used to develop several anti-cancer drugs, and some of them have been developed for cutaneous T-cell lymphoma and many other blood cancers, and they are also being looked into other dermatological diseases like skin cancers and even other skin diseases. So let's talk a little bit more about these epi drugs. So, like we have learned that uh, epigenetic dysregulation plays a causative role in diseases. So the answer must lie in modifying these regulators, right? So these drugs are based on these target enzymes, on the basis of their target enzymes or the regulators, like we have just studied. So based on these action, on the target enzymes, they have been classified into two types, which are DNA methylation inhibitors and histone deacetylase inhibitors. To understand the action of these inhibitors and what these enzymes do, let's take a look at the next picture a little bit. So, this picture might seem a little familiar to you because we have presented to you in the previous classes as well. So, look at this first picture. This is actually a nucleosome or a section of your chromosome or chromatin. So we can see some orange tags on the DNA, and these are acetyl tags. So like we have learned that when the DNA or chromatin is acetylated, it forms the euchromatin, and which is the active chromatin, which actively expresses and is transcriptionally active. So what DNMTs do, DNA methyltransferases do, is that they transfer methyl groups to the DNA and methylate the chromatin so that the gene can be silenced. Right? These red tags are the methyl groups which attach themselves to the nucleosomes, and they form and they silence the gene. And these H tags, histone deacetylases, they deacetylase the DNA, which means that they remove the acetyl tags, which tightly binds the chromatin, and it prevents the expression of the gene. It represses the gene, right? So now we know what DNMTs and H tags do. So how do you think these inhibitors could work? So what do you think the inhibitors would do to these enzymes? So take a look at this picture. So in this, the tumor suppressor gene is methylated. These red tags are methyl tags, which essentially switches off the gene. So now, because of the action of these DNA methyl inhibitors, the methyl tags are taken off or are removed from the gene, which switches it on, and it resumes its transcriptional activity. So these inhibitors are present during cell division when the cell actively replicates. So what this does is it prevents the DNMTs from transferring these methyl tags to the gene so that it cannot be methylated, and consequently the activity of the gene is not repressed. It can be transcriptionally active, and it can express its function easily. In a similar way, the histone deacetylase inhibitors also work in a similar way. They remove the acetyl tags, which causes the previously suppressed gene to express itself once more. Now, in the previous slide, I talked about drugs being developed for CTCL or cutaneous T-cell lymphoma. So, one of these drugs is known as vorinostat, and this works on the principle of high tags. So, right now we were talking about a lot of cancer therapy. So, this is one of the areas in which epigenetics is being studied very extensively. But it is also being studied in other areas because it is a very up and coming field of research, and it is providing scientists a lot of insight into how different physiological processes work and how treatments for other diseases and other processes could be found out. So, some of the current research that is going on is these are some of the examples of the research that is being done currently. So, before I move on to the next slide, I want to ask you guys if you know anything about these topics. So, have you heard of these terms, oxytocin, tuberculosis bacterium, or dementia? Do you guys have an idea about this? Okay, so we'll talk about these one by one. Right, so let's start with epigenetic uh, links to oxytocin. So, what do you guys know about oxytocin? Have you heard of this? So, what is oxytocin? Is it a protein? Is it what kind of molecule is oxytocin? Right. Yes, it is a hormone. And do you guys know any other names by which it is known? Yeah. So one of the names is also happy hormone. So oxytocin is commonly known by quite a few names, some of them being the love hormone, the happy hormone, and also cuddle hormone and birth hormone. So based on these names, you must have an idea of what oxytocin does in our body, right? It makes us feel happy. It induces happy emotions in us, right? So, scientists have studied um, oxytocin, and they have found out that this hormone is also linked to parental behavior and empathy. So, how do you think that epigenetics factors into parental behavior and empathy? Any guesses how epigenetics influences this hormone? Okay, so just answer this question if you can. That what do you think will higher levels of oxytocin do in our body? So, if there are two people, if you compare two people, one with low levels of oxytocin and one with high levels of oxytocin, how do you think they will behave? You can unmute and say if you have an answer. Yeah, Aviral.
I think the person with higher levels of oxytocin will be more happy or empathetic, probably.
Right. So, what scientists have observed is that uh, there is methylation in the oxytocin gene, and this epigenetic modification is linked with several personality traits and brain structure. Now, how this relates to maternal empathy or parental empathy and behavior is that a study was conducted among several mothers and women to see how methylation affects their behavior, methylation in their oxytocin gene. So, what it was found out that in those mothers in which there was higher levels of methylation in their oxytocin gene, they tended to be a bit harsher parents or less affectionate, less emotional with their children. And in those mothers in whom the oxytocin gene was lesser methylated, they were more openly affectionate or openly connected, empathetic with their children. So before I move on, I just want you to think about this. So how does this research, how does this factor into our day-to-day life? How can this help us? right? So just think about it and we'll discuss it a few minutes later. So let's come to the next one. So here is a picture, and I want you to answer what you think this represents. What do you think this picture represents? Any guesses? Yes, it's our brain, but there's an obvious difference. So why what do you think is this difference?
It compares the brain of a person with higher levels of oxytocin versus one with lower levels.
So it's not oxytocin. Instead, this is a comparison of the brain of a normal person and a person with severe Alzheimer's disease. So we know that Alzheimer's causes memory loss and a lot of other aging problems, but this is what the brain of a person with Alzheimer's actually looks like. It is severely damaged, as you can see. Now, Alzheimer's is something that we've all heard of, but there's no current cure for this disease. So a lot of research has been done into this. So what essentially causes Alzheimer's is that there are certain proteins which surround the brain cells and kill them, which leads to a loss of connection between neurons and which lead to these cells eventually dying. Now, this is what scientists have known up till this point, but now they have conducted a lot of research, and what they've found is that dementia, which is Alzheimer's, is a form of dementia, that a lot of a lot of genes regulate these diseases. So dementia is regulated by several genes, and they finally have some insight on how this gene regulation takes place. So a study was conducted, and it was found out that there are several sites throughout the genome of these brain cells where varying levels of methylation has been observed. In some places there has been observed very high levels of methylation, and some places there is low level of methylation. But these epigenetic modifications are pretty consistent in the brain of individuals with Alzheimer's, right? So, what do you think this research implies? How do you think this research can be utilized now that we know that epigenetic modifications also factor majorly into causing Alzheimer's? Can you guys guess or have an idea that how this research might help us? Right, so just think about it and we'll talk about it later. So let's come to the last article which I showed. So before I talk about it, again I have a picture for you and I need you to tell me what do you guys see in this. So this is, what do you think this is? Are these cells? Are these proteins? What are these? Okay, so I'll tell you what this is. So this is actually the bacteria which causes a severe lung disease, tuberculosis, and this bacterium is known as Mycobacterium tuberculosis. So what we have known up till this point is that this bacterium is a very growing microbe, but it develops antibiotic resistance extremely rapidly, right? So we know that organisms who have a shorter lifespan tend to mutate much more quickly, right? But in this case, it's a slow growing microbe, and even then, it develops drug resistance extremely rapidly. So scientists have asked this question many times, like how does this happen? And they have concluded that epigenetic modifications or epigenetic changes might be the reason as to why this bacterium develops resistance so quickly. So they have studied and found out that there is a type of DNA methylation called intercellular mosaic methylation. So what this methylation does is it leads to the bacteria developing many phenotypes or physical features like we had studied about in the first session. It diversifies, it creates many forms of itself even though it has the same exact genome. Now what this does is this helps it to adapt very quickly, adapt to the external environment, and it escapes the effect of antibiotics and it develops resistance against it. So, so this research has led to scientists finding new ways to treat tuberculosis, to find other ways instead of traditional antibiotics to treat this disease, which also becomes lethal at a point of time. Now let's talk about the implications of these research, how what this research implies and how it is being applied in real life now. Now we thus learned how oxytocin influences parental behavior and maternal empathy especially. Now many psychologists have noted this that harsher parenting leads to the maltreatment of children. So this research in which scientists have found out how this harsher treatment comes to be. Why do parents behave in such a way? So this research can help psychologists and other scientists find out how to maybe possibly reverse these changes and influence parenting. And this can lead to eventually a happier and more nurturing development of children, and it would prevent their maltreatment also. Like Aviral said, that this research in Alzheimer's could help in finding cures, and he said it rightly. So right now, so currently Alzheimer's has no cure, but now understanding how these epigenetic modifications influence the parts of brain, influence the cortex, so they can, so these changes can be reversed. So this provides a whole new insight into how drugs, drug therapy could be developed for Alzheimer's, which has never been done before, which is not prevalent in the society. So this is a very large breakthrough in the field of medicine, and it could possibly help a large number of people infected with this deadly disease. And lastly, for tuberculosis, like I said, that there are new mechanisms of killing this or tackling this bacteria are being developed. So, what scientists are trying to do is that they are trying to subdue or kill the bacteria before these epigenetic changes takes place, so that genetic mutation cannot takes place later. Because genetic mutations are mostly permanent mutations, like we're seeing in the COVID-19 virus. It is replicating so quickly and it is mutating extremely rapidly, and these mutations are permanent. So, but in this case, epigenetic mutations, if we stop the bacterium before they genetically mutate, we could possibly develop a new kind of cure for them. Right? So, I want you to ponder these questions and just think about how much this research actually influences the field of drug therapy. So, I'll pass it over to Sandhya now.
Hi, thank you, Ishani, for that. So, Moving on. So, we through this course, we've talked about what epigenetics is, why it happens, and um, what it's applied in now, the current work that's happening. And the natural next question is, where are we headed? As a scientific community, what is epigenetics going to do next, right? But because this is such a new field and it's so recent, there are a lot of fundamental questions that are still not answered. And in most cases, it is these questions that will lead the direction of the research because since they're so important, scientists will want to know the answer to those questions, and that will lead them to new research. So let's look at what some of these questions and see where we're headed. So the first set is mechanistic questions, which is the technical molecular details about what protein binds to what protein and how this interacts with DNA. It sounds a little boring, but let's just take a look. So the first is localization of heterochromatin. For those of you who remember, heterochromatin is the really tightly wound up DNA where gene expression cannot take place because the DNA is inaccessible. So if you can see the image that I'm pointing at now, um, here the heterochromatin, which is the dark blue structure, is located near the nuclear membrane. And on the microscope image on the right as well, this densely packed region of heterochromatin is located near the nuclear membrane. So this is a trend that's seen across all organisms. The question is, scientists don't know why this happens. They've tried inhibiting this localization by inhibiting proteins which facilitate this binding, and that has caused developmental disorders. But scientists don't know why. We know it's important, we know it's very important for survival, but we don't understand why yet. So that's a very important question that needs to be answered. Another one is the interplay between transcription factors and epigenetic modifications and how together they result in the gene regulation that we see. So, epigenetic modifications we've talked about a lot in detail, and that basically talks about how accessible the DNA is to facilitate gene expression. And transcription factors is a method we haven't explored that much in detail, but they're essentially proteins which bind to DNA under certain stimuli or under certain conditions. So they bind to the DNA and say, hey, look, this is accessible DNA that can be transcribed, or this is accessible DNA that needs to be transcribed. So we know that these two methods coexist in all of our cells, but we don't know how they interact. What genes are regulated by what? Is each gene regulated by both methods, or is it one or the other? We're not sure of that, and studying that is important because then we'll understand the regulation of specific genes, especially genes involved in diseases, a little bit better, and that will help treatment of the diseases eventually. The next is epigenetic triggers. We've been talking about epigenetic triggers quite a bit. So can someone tell me what epigenetic triggers are? There's not a formal definition, but just what you understand by the term. Okay, so epigenetic triggers is are any substances that cause epigenetic changes. It's fairly simple. So we've talked about in our application session last week, we spoke about different triggers and how they affect the epigenome. So we know that we have trigger, and trigger causes epigenetic change. So here a cigarette or smoking a cigarette will cause epigenetic modifications that make you more susceptible to lung cancer. So this seems fairly clear, right? What else is there to know? Turns out there's a lot. So what factors, what parameters of this relationship do we not know about yet? One is what all can act as epigenetic triggers. See, we know smoking, but we also know that environment and events that happen in our life can act as epigenetic triggers. So, is there something common that all of these hold which make them act like triggers? If there is, then that gives us a greater understanding of everything. So, what all can act as triggers, and how exactly do these act as triggers? Is it a particular molecule? Is it a reaction that our body has? What about it is a trigger for epigenetic changes? What is the time span we're looking at? Are we saying that I have to smoke 5 cigarettes a day for 5 years to notice a change? Or does one cigarette do the job? And if there is a change, what is the time scale for this change? These factors vary across triggers, and understanding them is important. And it's important because we cannot possibly analyze all the triggers, right? So if we can find patterns, then it will help us understand the overall picture in general. What is the pathway for these triggers? This might sound relatively unimportant, but molecular pathways are important because they are what form targets for drug therapy. So, only if I knew the specifics of what protein interacts with what part of DNA can I actually inhibit that interaction and thus prevent a disease or treat a disease. So why do our cells respond like this? We know we make epigenetic changes, but why? Is it an adaptive advantage or is it a defense mechanism or what is it? Why do we react like this? So, as much as studies have been carried out on epigenetic triggers, there's a lot we don't know which needs to be understood so that we can utilize epigenetics and the triggers to the best of our potential. So, this is a small video about epigenetics and where we're headed.
It's an exciting time to be studying this. Scientists are just beginning to understand how epigenetics could explain mechanisms of human development and aging, as well as the origins of cancer, heart disease, mental illness, addiction, and many other conditions. Meanwhile, new genome editing techniques are making it much easier to identify which epigenetic changes really matter for health and disease. Once we understand how our epigenome influences us, we might be able to influence it too.
So, like the video said, understanding this relationship is very important for us to be able to apply it in the best way possible. So, this was the mechanistic changes that I talked about were at a smaller cellular level, right? But these can be applied to bigger situations. So, to elaborate on that, I have one article that I read a few days ago, which I found very interesting, which I'm
It's
just
an
going
exciting
to show you. So, researchers find that regenerating the inner ear may be possible. So this sounds fancy. This sounds like I can grow a ear in the petri dish, but that's not what they're talking about here. That's also possible, but that's a whole different talk. So what this? Okay, let me explain what's happening here. So this blue cell is a supporting cell, and the brown cell is a hair cell. So as we all have studied in school, we can hear because of tiny little hair in our inner ear, which vibrate according to whatever we hear, and that's how we sense sound. So what happens is sometimes those hair get damaged. So when you're in your neonatal stage, that is during development before birth, um, supporting cells in your inner ear have the ability to differentiate to form hair cells. Right? That's how your hair cells develop in the first place. But as you become, once you're born, about a couple of weeks after you're born, your cells mature and they lose this ability. So your supporting cells, your blue cells, do no longer have the ability to become hair cells. And that's where the problem comes. That's why we all have relatives who, are hard of hearing. They can't hear. But this offers a possible solution to treating that. So what scientists did was they said, okay, we have two supporting cells and we see that there's a difference in their abilities, right? So what is their epigenetic difference? They checked that and they saw that in a neonatal stage, the histones of a few histones are methylated, while those were unmethylated in adults. So they call these methylation marks priming marks, right? So they said that these supporting cells are primed to be differentiated into hair cells, while in adult cells these are not. So the natural next step was to say, let's make sure that this is what's actually playing the most important role. And this is not just a coincidence that this epigenetic mark is present. So they inhibited the loss of this epigenetic mark. They inhibited the loss of the primer. And what they got were adult cells that had these epigenetic methylation marks and could differentiate into hair cells. So they've done it. They've realized that why adult cells cannot differentiate into hair cells is because of the absence of these epigenetic priming marks, as they call it. So this is just for the inner ear, but more and more scientists have found out that epigenetic modifications are what are responsible for preventing regeneration in general. You can cut off a finger and you won't grow back that finger. But we all know lizards can regenerate their tails. Starfish can regenerate one limb. So there are animals out there that can do it. And what majorly prevents humans from doing it is these epigenetic marks. And this is great for treatment because if someone loses hearing because of damage to these cells, you can modify the epigenetic marks in the other cells of the inner ear to let them regenerate their hair cells. This is all in very primary stages. This has only been carried out in mouse for now, but this is potential. And this is probably where we are heading in the future of epigenetics. So I this that was one small glimpse. But epigenetics in general is going to help us is answer questions in many fields. So give me your thoughts. What do you think? Scan this QR code and tell me, put in your thoughts about where you think the field is going. Where do you think it'll have a great impact? And it doesn't have to be epigenetics in general. It can be when you pair epigenetics with other technology like DNA editing technology, you get differences, you get impact, right? So, what fields do you think it might be able to impact? I have also put the link in the chat box so you can access it from there. Yeah, precisely. These are pretty much exactly the areas that. Oh, wait, we're getting more responses. Yeah, improving life, enhancements, medicine, disease treatment, cancer, they're all along the same line. So, yeah, as we've covered pretty extensively by now, um, disease treatment and medicine and healthcare is probably one of the most important areas where epigenetics will have an impact, right? And this is because of how much epigenetics has been implicated in disease development. So, a few points that I've put down are healthcare. So this helps not just in treatment, but in every step of the process. There's detection, there's diagnosis, there's treatment. All of these, if there are, say, epigenetic marks that are unique to populations that have a disease, then that helps in detection, diagnosis, and treatment for like epigenetic therapy, like Ishani talked about. Agriculture, someone said food, so that's essentially agriculture. If there are marks or epigenetic modifications that are unique to plants that have an advantageous trait, like higher yield or height in some conditions, then that might give rise to better, more fruitful plants. Livestock, which is also important. Epidemiology, here it involves disease treatment and prevention, but also it's not just about humans, right? When you're talking about epidemiology, it also involves the pathogen. So, like we spoke about last session, viruses and bacteria also have their own epigenome. So, studying that can help us understand viruses better, understand that better, so we can prevent, say, another pandemic or manage it better, probably. So, scientists all over the world are working on this because this is such an up and coming field, and because there's so much potential here because of how little we know about it right now. So, just a few examples. Dr. David Alice from the University of Rockefeller is one of the pioneers of this field. He has a lecture series which I have linked here below the picture that you can access and is great. It is not too hard to understand and it's very interesting. So, because he was one of the first people in the field, his research is more generic, like it's more widespread. He studies the general molecular mechanisms. And for anyone who might think that, epigenetics is this far-fetched thing that's done only in fancy labs abroad, it's not. In SNU, in Shiv Nara University, we have Dr. Sanjeev Galande, who is recently joined, and he studies epigenetic control a little more specifically in case of cancer and development and regeneration. So, for any of you who's particularly interested in regeneration, we are also hosting a winter course on it, uh, I think in December. So, do look out for that. Um, so yeah, so this if we're talking about where we're headed in the future, these are the names from whom we can expect that kind of research and development. So this is this is pretty much the end of the course in terms of content, but we have a quiz that's coming up. So just to brush things up a little before the quiz, we have a small recap. Before that, do you have any questions from what we just spoke about? Okay, great. Moving on. So, recap. So, what do we know, right? We know there are epigenetic modifications, and we know what epigenetics is, right? It's changes beyond the primary DNA genetic sequence. And broadly, if you had to classify it, it can be of two types. Either based on outcome, this is so epigenetic modifications can either cause gene expression or they can silence gene expression. So, causing gene expression means that I will have to make my DNA more accessible. So, that'll have to be loosely bound. So, working backwards, if it has to be loosely bound, then my DNA and histones should not interact as well. So, my histone should become less positive. So, it should be acetylated. I'm sorry, this is a mistake. This should be acetylated. It should be histone acetylation, which makes it less positive and loosely bound, causing gene expression. Then the second category is gene silencing, which is a little more elaborate. So, gene silencing, the primary thing of gene silencing is heterochromatin or chromatin that's really tightly wound together. The first easiest way to do that is histone methylation, which is making histones more positive so they bind tightly together. The next is DNA methylation, which we spoke about, I think, in session two, which is DNA binding, methyl binding to the DNA. So your polymerase or your enzyme that makes DNA protein cannot bind to your DNA. So that's DNA methylation. Again, it silences genes. And your final method is RNA mediated silencing, which we saw an example of in X inactivation, where a single stranded RNA binds to another single stranded RNA or DNA, and the process stops there, right? You don't get a resultant protein. So that was a quick, short recap. And again, if any of you have any doubts for pretty much anything across the course, all of us are here, so we might be able to answer your questions. Okay, then. Um, so now we have the quiz, and the link is in the chat box, so you can all join. Okay, so I think all of you have joined, so I'm starting the quiz. Okay, now this is your last question. Okay, that was the end of the quiz. Shruti will take over now.
Uh,
Hi guys, just
Sandhya, I think Amiral has a question. Sorry.
look into the last question and we'll get back to you for that. Is that all right? Okay, so thank you guys for joining in. We'll look into the last question as you guys, as someone has requested. Also, so this was the end of our 4 session certificate course. And if you have any other questions, please hit us up. So as you know, this event has been co-hosted by Genesis and KaryoUS. And about Genesis, again, we're the departmental society of Shivnati University. And we at university we do hold a lot of similar events for the students, and a lot of times we also do outreach programs as such. And we hope to see more of you coming soon. Feel free to follow us on our Instagram page. Here's just a small screenshot of what it looks like, where we post a lot of frequent facts and fun information about everyday science, such as we did biology and cooking, and something we think you guys might be interested in. Also about KaryoUS, it's a wonderful initiative taken up by Vinita ma'am and her team. And please do visit their website. And they have been holding a lot of certification courses and outreach courses as well for students within the Shivnadu community and outside as well. So please do follow all of us. And again, on the KaryoUS website, we have all our links, and you can access all our videos and have a look at more upcoming sessions that take place. So thank you for tuning in. I would like to call upon Sandhya to give the thank you note, and yeah.
Also, Janvi, all the students, before they leave, Mala, would you like to announce after Sandhya? Please announce that any feedback or anything can be shared and whatever. Over to you, Sandhya.
Yeah. Sandhya, you there? Okay.
Sorry, yeah, I'm here. Sorry.
Yeah. Okay, so this was the final session, and I hope all of you enjoyed, going through all these sessions with us. It was a huge learning curve for all the children, and it was really fun, working together for many of our university students with the school students with us. So I think I have at least enjoyed working through all of this. And in the future, we would for future events that we might want to plan and hold, we would expect feedback from all of you, those who have participated in this, so that we can just improve upon this and bring such events to you in a format that would be enjoyable by all. And you will also benefit from the learning experience of not just presenting the content, but also, just organizing events like these, because it's not just the biological content that which is at which is being kind of planned and tested. It's also about organizing stuff like this, bringing people together for a common cause. So I just hope all of you enjoyed this, and it was an amazing job done by all of you, whether it's the SNU students who planned and presented it, whether the school students who worked with them and they benefited from the knowledge and from the skills that the seniors have. And this is also an exposition for the school students to look forward to what is going on in the college level. So it helps them also think about biology and what are the careers possible available there. So it's an amazing opportunity for you guys to be the mentors to them. So it's like passing on the legacy of the science, the science as an enterprise, to the school students as well. And it's for all of you, not just the students who are presenting, but even many of you who have participated in this. Everybody has contributed in some way or the other to this um, the growth for all of us, right? So we will wait for your feedback. All of you, just do write to either Genesis or to KaryoUS, whoever you feel comfortable writing. And amazing job, Sandhya, Janvi, Ishani, Bhuvan, Kartavya, and I'm just anybody who I may have missed out on. And then the school kids, Priyanshi, Arushi, and Suhana, they have done such an amazing job at their end, despite being such young students. So all the best. Sandhya, would you like to just close it here?
Um, yeah, sure. So, yeah, nothing new, honestly. I just wanted to say thank you to everybody involved. Um, Thank you for attending these sessions with us. We really hope that you learned something. Like Mala ma'am said, we've learned this has been so much more than just subject for us. It's been learning how to organize something like this, how to carry it out. Again, I'd like to thank the whole team that was part of this, everyone who worked on putting this course together. Our student mentors, that's Bhuvan, Ishani, Kartavya, our school students who worked with us, Arashi, Suhana, and Priyanshi, who really were there at every meeting, every time we met and discussed things, who contributed a lot to the presentations as well. Our mentors, Vinita ma'am, of course, Mala ma'am, Ruchi ma'am, and Chetna ma'am. Also at SNU, Richa ma'am and Anandita ma'am, who helped with designing content and sat through our dry runs and met us at 10 minutes notice. Thank you so much. Also, thank you to Janvi and Shruti from Genesis who put in a lot of time and effort at the back end so that this whole thing ran smoothly for us. And most importantly, thank you to you guys for attending these sessions because if you didn't attend, we would not have a course to create and give you. So thank you so much for your interaction, all the questions that you've asked and everything. Um, and for those of you who attended most of the sessions, you will receive a certificate from Genesis. You'll receive a certificate on your email soon. Don't worry about that. We will also send you a feedback form that you can fill in and feel free to reach out to us at any point if you have anything that you'd like to discuss. For the last time, I'm Sandhya. I'm signing off on behalf of Team KaryoUS and Genesis. Thank you once again.
Yeah, one thing I would like to add. All the participants, would you like to switch your videos on? Just say one sentence or just one or two lines to our presenters. Would you like to say something about your experience? Just one or two lines.
Mala ma'am, even I would like to speak about, but I'm
Yeah,
sorry,
Shriya
my
Vanita, please
video
go
is on.
ahead. Please go ahead.
So, I have never seen such a beautiful collaboration between school and college kids. And there are no words to say thank you to these children who, came forward and they said that yes, they can take it. So hats off to Team Genesis, hats off to Team KaryoUS. Special thanks to Sandhya who first took session on epigenetics, gene CRISPR, few months back with Dr. Koeli Mappa and mentored by Mala ma'am. And it all started from there actually. And Janvi, you don't require to do MBA. You are already MBA graduate. I can see the way you are managing things. And all of you, Bhuvan, Ishani, and the school children. I mean, I'm completely speechless, Mala ma'am. The way these kids have carried out with so much of grace, so much
Yes.
of grace, and the way they took it. I mean, I would like to see this video specifically, the last session of this particular thing when you guys are speaking again and again. I want to show this that this is called real collaboration where seniors are hand holding the juniors and the way it is taking forward the legacy of SMU along with SNS and Vidyaga.
Yes.
Thanks once again.
Yes. So and I see that some of you are just naturals. When I saw Ishani and Bhuvan and Sandhya, everybody presenting to people, I feel that you are so naturally involved in it, and you're so natural in connecting with the audience and everybody in planning and presenting. It's really, and we do need people like you, to uh, take science to the school and the other children. So, from our audience, for the participants, who have been together with us over the past 4 weeks, would you like to say something? Just, one or two lines. The floor is open to all the participants. Anyone?
I just wanted to say that this was the like most interesting course ever
Uh
I
huh.
ever attended, and like I am new to
I think
I
sis
think there
has.
is some problem. So
I enjoyed the course a lot too.
Uh-huh.
in chat box, yeah, can see he can
Yeah.
he or she can express his feeling in chat box or can write mail also. Mala.
Yes. Chal.